Hello, folks. This is Tolbert's class, biology. This is lesson. This is the week two lesson. So here we go. Um, there's some information here that you'll need to see. Um, just some things to think about. The DNA molecule must somehow specifically enable these proteins to be made and regulated through various functions of the cell. What kind of structure could serve this purpose for carrying this out? Understanding the purpose or the structure of DNA is key to understanding how genes work. DNA. DNA is a nucleic acid made up of nucleotides. This is what you need to know. It's a nucleotide joined together by long strands of covalent bonds. Nucleic acids are very long. Slightly acidic molecules originally identified as in the cell nuclei. Nucleic acids are made up of nucleotides linked together to form long chains. So similar to like when we were talking about proteins, in proteins, the building blocks of proteins were amino acids, and these amino acids all linked together to make a protein. In nucleic acids, these specific nucleotides all linked together. Each nucleotide is one link in the chain, and they make up our nucleic acids, which makes up our DNA. Nucleotides. So these are what these kind of look like. They come from these base information that we should already have seen before, but these nucleotides are known as adenine, guanine, cytosine, and thymine. A, G, C, T. You need to know that every single nucleotide is composed of some basic parts. Every single nucleotide has a base, like adenine, guanine, cytosine, and thymine. And every nucleotide has a sugar called deoxyribose. And finally, every nucleotide has a phosphate group. DNA's nucleotides are made up of the three basic components: a five carbon sugar called deoxyribose, a phosphate group, and a nitrogenous base. Nucleotides and a strand of DNA are joined by covalent bonds formed between their sugar and their phosphate groups. You might remember covalent. Covalent means shared, sharing electrons. Carrying, sharing is covalent. Covalent bonds are strong. DNA has four kinds of nitrogenous bases. As we've already stated, the A, T, G, and the C: adenine, guanine, cytosine, thymine. The nucleotides can be joined together in any order, any way, any order to make these sequences. It's just the arrangement of these sequences that allows it to have all of the unique diversity that we see in DNA today. What clues help scientists solve for the structure of DNA? We've heard about Miss Franklin. Miss Franklin's X-rays patterns enabled Watson and Crick to build a model that explained the structure of and the properties of DNA. Here's another one, Chargas rule. Chargas rule has to deal with whenever you have a letter or one of these exact base pairs, you're going to have exactly the other one on the other side: A to the T, C to the G. So his rules basically says the percentages of adenine and thymine bases are almost equal in a sample of DNA. The same thing is true for the other two nucleotides, that is, guanine and cytosine. The observation that A is equal to the number of T and G is equal to the number C is known as Chargas rules. Here's another way that we can think about that. Also, this is a graphic organizer, so if you'd like to write this down, this is a great way for you to remember for you to understand how DNA is kind of organized. You're going to have the exact same amount of adenine as you have thymine. It just so happens that adenine and thymine are the ones that are base paired together. Guanine and cytosine, they're going to be base paired together as well. In 1950, British scientist Mrs. Rosalind Franklin used a unique technique called X-ray diffraction to get information about the structure of this DNA molecule. It revealed an X-shaped pattern showing that the strands in the DNA are twisted around each other like coils of a spring. The angle of the X-shaped pattern suggested that there are two strands in the structure. Other clues suggest that the nitrogenous base near the center of the DNA molecule. At the same time, these guys, kind of criminalistic, had a guy. These guys were coming close to the understanding, but they didn't know. Okay, they built some three-dimensional models, but it wasn't until they got information from Miss Rosalind that actually allowed them to build the proper model of DNA. In early 1953, Watson was shown a copy of Franklin's X-ray pattern. The clues in Franklin's X-ray pattern enabled Watson and Crick to build a model that explained the specific structure and properties of DNA. Watson and Crick's breakthrough model of the double helix, which was two strand wound together, which was what they got the Nobel Prize for. Thank you.